So now we're going to dive into the cell and take a look around at all of the different organelles and structures that we find inside of a cell. The cell is called the fundamental unit of life, and that is because they are the simplest structure that can be considered living. So all of those properties of life that we require in order to call something alive, a cell is the smallest thing that can do that. So there are several tenets of what we call the cell theory, and one of those is that all organisms have to be made up of cells. So in order to be considered a living organism, you have to be made of at least one, if not more, cells. That cells are the smallest thing that can be living, and all cells descend from earlier cells. So what we're going to see is that in order to make new cells, you start with one cell, and then it splits into 2 or 4, depending on what kinds of cells you're making. That probably begs the question, like, well, where did the first cell come from? And we have some theories about how maybe we might have developed early cells, but those are going to be saved for biology too. So in general, what we're going to see is that one cell becomes more cells, and we don't just randomly, spontaneously generate cells from non-living matter. So we can't talk about cells and the structures inside of cells without talking about the way that we study them. Cellular biology is fairly new within the last century or so because of the development of a tool called the microscope. So the microscope was the first scientific tool that allowed us to actually look directly at a cell. And the first person to name some cells was Robert Hooke, who looked at some cork, and he thought that all of the spaces in the cork is dead plant matter from bark of a tree, and he saw what was the remnant of cells, and he called them cellulae, or little rooms, and so that's how cells got their name. So the first type of microscope that was discovered was a light microscope, and what this does is it sends visible light through a specimen, so you can put some cells on a glass slide, and you can send light through them. And then what you do is you look through a lens, and a lens is a curved piece of glass, and that curve to the glass refracts, it bends the light, and then the image is magnified. So early light microscopes only had one glass. Nowadays, most often they have multiple lenses, and you can change the lenses to increase the magnification. When you increase the magnification of an image, the resolution decreases. So this is like taking a picture and zooming in. That's magnifying. And when you do that, the picture gets grainier and grainier. And if you zoom in too far, the resolution is too low to see anything. You just see pixels. And that's what happened with these first early light microscopes, is the resolution on these is too low to study most of our organelles. The big organelles like the nucleus, those are visible typically in light microscopy, but some of the smaller organelles, the resolution is just too small and it's not going to be able to see them. Over here you can see the range for the light microscope, and this is the log scale, so each big increase is as a increase of times 10. And so the light microscope, the smallest it can usually see is down to the order of microns, which is micrometers here. You're going to be able to see a nucleus, some small bacterial cells, maybe some of the other larger organelles at that point. Usually these are really high-powered light microscopes. They're going to require oil in order to capture that image. Most light microscopes are going to be over here, where you can see to the size of most cells, large microorganisms. There's a little bit of an overlap here of what you can see in a light microscope with what's visible, some smaller things that are visible at the unaided human eye. Up here you can see an image of some blood. So these are red blood cells, and then this is a white blood cell, and this is a light microscope image. So it gives us a nice image of what the cell looks like, and you can see somewhat of the inside, the granules, the nucleus inside of this white blood cell, but you really can't see much more than just kind of the shape of the cells and where they are in relation to each other. So how do we study these really, really small structures within a cell? How do we actually visualize those? There are different types of microscopes. So an electron microscope is similar, except instead of using light, it uses electrons. So it will shoot a beam of electrons at a object. And then that beam either does one of two things depending on the type of electron microscope. A scanning electron microscope, the beam is going to bounce off of the surface and it provides what looks like a 3D image. So up here, you can see a scanning electron microscope image of, again, red and white blood cells. Here's the red blood cells. And this is a lot better resolution. Instead of just seeing some circles now, you can actually see the discoid shape. You can see that indentation in the center. And then you can see the white blood cells and you can see how grainy they look. But what you can't see is you can't see inside the cell at all. You're only seeing the outer surface. And then there is another type of electron microscope, and this is the transmission electron microscope. And what this one is, it focuses a beam of electrons through a specimen. So in this one, what you're going to see is you're going to see the cross-section and look at the internal structures rather than the surface. So this is a capillary here, which is the smallest arteries in your body. And this is actually one red blood cell, one single red blood cell, which is about 7 nanometers large, which is actually bent because your capillaries are so small that the little red blood cells actually have to bend to go through them single file. And so you can see the massive difference in scale that these scanning and the transmission electron microscopes especially they have great resolution to be able to see really, really tiny things. The problem with these types of microscopy, the preparation of the specimens damages the specimens. So you cannot see living specimens. If I want to study red blood cells that are alive and watch them actually moving, I need to use light microscopes. Light microscopes don't harm the actual specimen. Scanning electron and transmission electron, in order to get the beam of electrons to interact with the specimen, you have to actually damage and kill the specimen to prepare it for these microscopes. So it's a trade-off between resolution and being able to see these tiny, tiny things versus being able to see complex processes that are actively working. Another method of early ways to study cells was cell fractionation, and this was a method of systematically taking cells apart from large molecules to smaller and smaller molecules. And that helps us figure out the functionality of some of these tiny, tiny molecules without the ability to be able to see them. So what ends up happening here is this is a sample of tissue cells. They get homogenized, basically they're blended up. And what that does is it breaks apart all of the cells and lets all of the components come together. The centrifuge will spin very fast. It's going to send the most dense and heaviest materials to the ends, and then the small materials will be in what's called the supernatant. So this pellet at the bottom, after it's centrifuged, is going to be full of really large cell debris, the really large nuclei, and the less dense material will be floating up here. Then we pour off the top and centrifuge it again. And so each time it goes through the centrifuge, the pellet at the bottom is consisting of smaller and smaller and smaller organelles. So what you can do is you can pour off until you basically this supernatant that's full of teeny tiny molecules like ribosomes, and you can actually use this fluid to study those specific components or